Hello students, I welcome all of you to Edukemy's class of Science and Technology. आज के क्लास में we are going to study a very important topic regarding biotechnology. So the component that we'll study today is biotechnology, and biotechnology का सबसे important and सबसे पहला lecture होता है regarding DNA basics. So आपको समझना है, you'll have to understand that why DNA is at the central stage of biotechnology. Now we study, this is the first component of your science and technology, that is the biotechnology, जहाँ पर more or less 4 to 5 questions come every year, from biotechnology. Now you'll have to understand that all of the biotechnology actually revolves around DNA. Whether it be Human Genome Project, whether it be gene editing, whether it be 3-parent baby, whether it be designer baby, whether it be mitochondrial donation, whether it be induced pluripotent stem cells, whether it be Earth Biogenome Project, fine. Whether it be all of these important projects, 100,000 Genome Asia Project, fine, Chimpanzee Genome Project, all of these projects are actually, the central stage of all of these projects is DNA, fine. So in this particular lecture, we are going to understand what is the DNA basics, how the DNA is constructed, what are the bases, what are the base pairs, fine, what is a nucleoside and what is a nucleotide and what is a nucleic acid, and then the difference between DNA and RNA, and how we actually construct chromosomes from DNA, and what is the central dogma of molecular biology. Now this is our today's lecture regarding DNA basics. Now you'll have to understand that biotechnology will give you almost 4 to 5 questions in your preliminary, so you'll have to be very attentive in the class to understand the DNA basics. Now see guys, in our cells we all know there is a nucleus, right? There is a nucleus, and we have 5 nitrogenous bases in our nucleus. How many? 5 nitrogenous bases. 5 nitrogenous bases. There are 5 nitrogenous bases in our nucleus of a cell. Now, what are these 5 nitrogenous bases? People that are not from the science background, these are some very simple words. You need not to get into more detail. Fine, this is UPSC that we are talking about. We are not actually going for a particular science subject, or we don't have to actually crack a particular science exam. This is UPSC, and UPSC actually asks what is required for general awareness. We want people that are actually generally aware of things, fine. So this is one of those things that you should be aware of, fine, because you have to be an informed citizen, and to be that, you need to know what DNA basics are. Now, in our nucleus of a cell, there are 5 nitrogenous bases. What are these bases? The bases are known as maybe adenine. Adenine is a base. Adenine is a base, and it is a nitrogenous base. Nitrogenous base means it has nitrogen in it. Fine. So it is a base. You have that chapter in your 9th class in CRT, whereas there's a chapter on acids and bases. So it is a base. It is a nitrogen containing base. That is why it is known as nitrogenous bases. Okay, nitrogen, we know the most abundant element in our atmosphere is nitrogen, and in our human body also nitrogen is actually in large concentrations in our body as well. Now this is your first base, that is your adenine. Then we have one more nitrogenous base in our body, that is known as thiamine. You'll have to remember these names. It will be very important and very beneficial for you to remember these names. Adenine ke baad we have guanine. Guanine is another nitrogenous base. Then we have cytosine. And then we have uracil. Now, if anyone tells you what are the 5 nitrogenous bases in our cell, that is adenine, thiamine, guanine, cytosine, and uracil. These are the 5 nitrogenous bases. Now, when we add some things to these nitrogenous bases, they actually change to something else. Fine. Now, these nitrogenous bases, let me clear this up. Now, I want actually to have a lot of space here to construct DNA. What we are studying? We are studying DNA basics. Okay. Now, we have 5 nitrogenous bases: adenine, thiamine, guanine, cytosine, and uracil. Okay. Now, when these 5 nitrogenous bases, when we add a particular component in here, it will actually change to something else. Now, when we add sugar to these bases, when we add sugar to these bases, it gives us nucleoside. It gives us nucleoside. So nucleoside is nucleoside is made from base and sugar. Base and sugar. So what we have nucleoside. And when we have nucleoside, we add a particular component in here, that is phosphate. Okay. Now आपके पास बेस है, बेस आपको मिल गया, आपके नाइट्रोजनस बेसिस जो ऑलरेडी होती है उसको आपने क्या दे दिया, शुगर दे दिया, आपको क्या मिला, यू गॉट न्यूक्लियोसाइड. न्यूक्लियोसाइड को आपने प्लस किया फॉस्फेट, इन दोनों से मिलकर व्हाट डिड यू गेट, यू गॉट न्यूक्लियोटाइड्स. नाउ बेस प्लस शुगर न्यूक्लियोसाइड, न्यूक्लियोसाइड प्लस फॉस्फेट न्यूक्लियोटाइड. अब न्यूक्लियोटाइड आपको मिल गया. न्यूक्लियोटाइड का अगर आपके पास चेन है, इफ यू हैव अ चेन ऑफ न्यूक्लियोटाइड्स, that will give you nucleic acid. So this is what nucleic acid is. Nucleic acid is a chain of nucleotides. What is nucleotide? It is made from phosphate and nucleoside. What is nucleoside? It is made from a base and a sugar. Fine. Now when you have nucleic acid, these nucleic acids can be of two types. One is deoxyribonucleic acid and other is ribose nucleic acid, and that is your DNA and RNA. Okay. So let us see that in a diagrammatical representation. Now before we see that, we need to know what this DNA and RNA is. Okay. Now see guys, we generally say, this is not the class in which we can go into more detail between DNA and RNA. Let me give you some of the examples. Okay. Now DNA, in this DNA, we generally say it has a double helix structure. Double helix structure. All of you know that it is something like this, right? It is known as a double helix structure. So DNA has a double helix structure and RNA is single-stranded. So this is the basic difference between DNA and RNA. DNA has a shape of double helix and RNA is single-stranded. RNA is something like this, okay? It is not double helix. It has not double helix structure. उसके बाद DNA में आपने कितनी बेसिस यहाँ पे रखी? एडेनिन, थायमीन, गुआनिन, साइटोसिन एंड यूरेसिल. अब यहाँ पर 5 bases थी, but DNA is only made from 4 bases, and that is adenine, thiamine, guanine, cytosine. But uracil is absent. Uracil is absent in DNA. But in here RNA में uracil is present. Okay. So that is the difference between DNA and RNA. Uracil is absent in DNA. Uracil is present in RNA. Okay. Now ये जो मैंने यहाँ पर लिखा, सेम इसी चीज को हम क्या करेंगे? We will construct it with the help of a diagram. So diagram से चीजें क्लियर होंगी, वहां से ही आपको समझ आएगा DNA कैसे बनता है. Okay. So since we are talking about DNA basics here, we will only talk about DNA in this class. Now सबसे पहले what did you have? You had 5 nitrogenous bases. Okay. So in 5 nitrogenous bases में, since we are actually constructing DNA, we actually have to include only 4. Because I already told you DNA के अंदर सिर्फ 4 bases होती है. That is adenine, thiamine, guanine and cytosine. Uracil is absent in DNA. So in 4 nitrogenous bases में से, let's take some random base. For example, we have here adenine. So I took a base, that is adenine. उसके बाद 5 nitrogenous bases को plus क्या करना है? Sugar. So let us add sugar in here. तो मैंने यहाँ पर sugar add कर लिया, and this is your sugar. अब ये दो मिलकर क्या देंगे? This is a nucleoside. Okay. तो यहाँ पर आपको nucleoside मिल गया. That is your nucleoside. फिर nucleoside को you will have to add phosphate. So we'll be adding phosphate in here. तो अब यहाँ पर हम phosphate इसको add करेंगे. Suppose we added phosphate. Okay. Now this particular structure is of a nucleotide. ये आपको मिलकर क्या दे रहा है? Nucleotide. This is your nucleotide. So we had nitrogenous base plus sugar, that gave us nucleoside, then phosphate, and that gave us nucleotide. So this is a particular nucleotide. And then I told you, you need a chain of nucleotides. So let us make a chain of nucleotides. So we'll have another base, कोई और बेस उठा के देख लो, सपोज थायमीन, सो दिस इज अ थायमीन और उसका शुगर और उसका फॉस्फेट, ओके, दैट इज अगेन अ न्यूक्लियोटाइड। अब न्यूक्लियोटाइड की चेन बनाई जा रही है, देन यू हैव अनदर शुगर, अनदर फॉस्फेट एंड अनदर बेस, सपोज साइटोसिन, देन यू हैव अनदर शुगर, अनदर फॉस्फेट, अनदर बेस, सपोज दैट इज guanine, that is guanine, okay. Now you already have a chain of nucleoside. This chain is known as a nucleic acid. This is known as nucleic acid. This is known as nucleic acid. But in this class, we are concerned with deoxyribonucleic acid, that is DNA. That is DNA. And I told you, DNA is double helix. DNA has a double helix structure. And how do we construct double helix structure in DNA? Because in DNA, bases are able to join with each other. They are able to make weak hydrogen bonds with each other, okay? So let us make a DNA. This is a chain of chain of nucleotides. It is a nucleic acid, but we want deoxyribonucleic acid, so we'll be taking some other bases. Now we have to understand that these bases make bonds with each other. Adenine base makes a bond with thiamine base, and guanine base makes a bond with cytosine base, okay? They make bonds with each other. These are weak bonds, but they do make bonds with each other. So we have adenine base, it will make a bond with thiamine base. And when we have a thiamine base, it will have its sugar, it will have its phosphate, okay? Then we have thiamine. Thiamine will make a bond with adenine. So thiamine will make a bond with adenine. Adenine will have its sugar, adenine will have its phosphate, okay? Then we have cytosine in here. Cytosine is making bond with guanine. So let's make a bond with guanine, and this is a sugar, this is your phosphate. Then you have guanine. Guanine will make bond with cytosine. Let's make a bond with cytosine, uska sugar or uska phosphate. Now this is your DNA, right? यही आपका DNA construct किया जा रहा है. Now that is your DNA, and this is known as your base sequence. Your base sequence. What is the sequence of the bases? The sequence of the base is TAGC. TAGC. What is the sequence? TAGC. This is known as DNA base sequence. This is known as DNA base sequence. उसके बाद these two bases make a pair. These two bases make a pair. That is known as base pairing. That is known as base pairing. Now you see, with base pairing, the sequence that you have from the lower side, you will have exactly opposite sequence from the upper side. तो अगर यहाँ पे T है, यहाँ पर A होगा. अगर यहाँ पे A है, यहाँ पे T होगा. अगर यहाँ पे G है, यहाँ पे C होगा. अगर यहाँ पे C है, यहाँ पर G होगा. Exactly opposite होगा. Fine. Now you can see यहाँ पर exactly opposite bases है. Now this is your complementary base pairing. This is your complementary base pairing. That means नीचे से जो आपकी base है, उसका exactly opposite आपका ऊपर से होता है. Fine. Why? Because they make bases, they make weak bonds with each other. Okay. Now we have understood how the DNA is made. How the DNA is made. Now from this DNA, you need to understand how do we construct chromosomes. Now before we actually construct chromosomes, I want to give you some information about DNA. Now see guys, why is actually the entire modern biotechnology, it revolves around DNA. All of you, suppose coronavirus हो रहा है, coronavirus में we got to know about the genome sequence of coronavirus, जो एक RNA virus है, okay? या RNA virus में जो वैक्सीन हमने बनाया है, suppose mRNA वैक्सीन आपने सुना होगा, ये mRNA वैक्सीन कैसे निकाला? हमने जो है कोरोना वायरस लिया, उसकी बेस की सीक्वेंस निकाली, ये सीक्वेंस निकाली उसकी. बट कोरोना वायरस में इट इज नॉट अ डीएनए, उसका जेनेटिक मटेरियल डीएनए में नहीं है, उसका जेनेटिक मटेरियल आरएनए में है. ओके, नाउ यही सीक्वेंस जो है, दिस इज नोन एज इंफॉर्मेशन. नाउ सी गाइस, सपोज let me give you an example. When you actually make and develop a software, software को develop करने के लिए you need to have a code. आपके पास एक कोड होना चाहिए. जब कोडिंग करते हैं, पूरा का पूरा प्रोग्राम बनकर आता है, राइट? बिल्कुल ऐसे ही हमारी बॉडी भी एक कोडेड फॉर्मेट में है. हमारी बॉडी को जब बनाया गया, उसमें भी एक कोड पहले लिखा गया, उसी कोड के हिसाब से हमारी बॉडी बनी है. फाइन. और यही वो कोड है, दिस सीक्वेंस दैट वी हैव, TAGCACCCTT करके, ये तो लंबा डीएनए का सीक्वेंस है. दिस इज द कोड ऑफ योर बॉडी. इसी कोड के हिसाब से यू हैव गॉट द कैरेक्टरिस्टिक्स दैट यू हैव। आपकी बॉडी के सारे कैरेक्टरिस्टिक्स, आपका बिहेवियर, आपकी बॉडी, आपका इम्यून रिस्पॉन्स, योर सेल्स, योर लुक्स, योर फिजिकल कैरेक्टरिस्टिक्स, all of them depend on this code जो आपकी सीक्वेंस बनाता है डीएनए के अंदर। नाउ सी गाइस, हमारी बॉडी में भी बिल्कुल कोडिंग हुई है जिसकी वजह से हमारी बॉडी कंस्ट्रक्ट हो पाई है। नाउ सी गाइस, you need to understand this concept here. For example, let me give you an example. Now see, आपने पढ़ा होगा this is our body, बचपन में पढ़ा होगा। बॉडी इज एक्चुअली एन ऑर्गन सिस्टम। बहुत अलग अलग ऑर्गन्स मिलकर एक पूरी बॉडी बनाते हैं, राइट? दैट इज एक्चुअली एन ऑर्गन सिस्टम, राइट? इट इज एन ऑर्गन सिस्टम। सो बॉडी इज एक्चुअली एन ऑर्गन सिस्टम। बट ये बॉडी कैसे बनती है? बॉडी बनती है डिफरेंट ऑर्गन्स की कॉम्बिनेशन से। सो एक्चुअली बॉडी इज मेड फ्रॉम ऑर्गन्स, व्हेन यू हैव एन एंटायर ऑर्गन सिस्टम, then we call it a body. Okay, so body is actually made from organs. तो organs कहां से बनाए जाते हैं? Organs are made from tissues. तो आपको organ चाहिए, you need an organ. For example, liver is made from liver tissues. Heart is made from heart tissues. Fine, कोई भी organ उठाओ, it is made from a tissue. उसके बाद these tissues are made from cells. Okay, now when you have particular set of cells that combine together, then we have a tissue. एक particular type of tissues, they come and make organs, and different organs come and make the body organ system. Then these cells are actually made from proteins. Whenever we actually code, जब मैंने आपको कहा सॉफ्टवेयर डेवलपमेंट में जब हम कोड बनाते हैं, वो कोड असल में आपका जब सॉफ्टवेयर बना के देता है, वो असल में आपका प्रोटीन होता है। क्या होता है? प्रोटीन की वजह से ही सेल्स बनती है, टिश्यूज एंड ऑर्गन्स एंड ऑर्गन सिस्टम। दिस इज वेरी इंपॉर्टेंट फॉर यू। प्रोटीन्स अगर नहीं होंगे, बॉडी वुड नॉट हैव फॉर्म्ड। फाइन, प्रोटीन्स आर वेरी इंपॉर्टेंट, एंड प्रोटीन्स आर मेड फ्रॉम अमीनो एसिड्स। They are made from amino acids. When you have a chain of amino acids, that is known as a protein. You have amino acids, and when these amino acids combine and make a chain, this chain of amino acids is known as protein. That is a protein. And these amino acids, you know, how do they combine? I told you amino acids, these are different amino acids. When these different amino acids come and combine and make a chain, that gives you protein. So this is actually a protein chain. Which is actually a chain of amino acids. Okay, so you have a chain of amino acids that create a protein, and different combination gives you different proteins. Okay, now how do these amino acids know that in what form they have to actually combine and make a chain? How do they know that what is going to be the sequence of the chain? How should these amino acids combine? That information is coded in DNA. मतलब क्या है इसका? DNA आपको बोलेगा, DNA अमीनो एसिड्स को बोलेगा आपको कैसे कंबाइन होना है ताकि वो क्या बना सके? प्रोटीन बना सके. So DNA has the information that how should the amino acids combine to form the proteins, and when you have proteins, then you have the entire system. Okay, so this information is actually present in the DNA of the body, and DNA has that coding जो आपको बताएगा amino acids को how do you have to combine. Okay, now let me give you a more precise example of this system. Now see guys, this information is actually present in let me make a big diagram. This information is actually present in the nucleus because DNA is inside the nucleus, right? This is your nucleus and this is your DNA. This is your DNA, okay? This is your DNA. Now I told you this DNA has the information how the amino acids have to combine to form the protein. Now you have to understand कि अमीनो एसिड्स को कंबाइन होना है, बट वेयर आर द अमीनो एसिड्स? अमीनो एसिड्स यहां पर होते हैं साइटोप्लाज्म में. कहां पर? साइटोप्लाज्म में आपके अमीनो एसिड्स होते हैं. सो यू हैव अमीनो एसिड्स हियर, you have amino acids there. Now these are the amino acids, but these amino acids do not know how do we have to combine. प्रोटीन बनाने के लिए हमें कॉम्बिनेशन कौन सी सीक्वेंस में करना है इनको नहीं पता. वो किसको पता है? डीएनए को पता है. अब डीएनए में इंफॉर्मेशन है कि अमीनो एसिड्स को कंबाइन कैसे होना है, पर डीएनए तो अंदर है न्यूक्लियस के, अमीनो एसिड तो बाहर है न्यूक्लियस के, तो ये इंफॉर्मेशन अंदर से बाहर कैसे जाएगी? How do we carry this information from this nucleus, from inside this nucleus to the cytoplasm, ताकि हम अमीनो एसिड को बता सके how do you have to combine. Now you have to understand this information is carried by RNA. This information is carried by RNA because RNA सिंगल स्ट्रैंडेड होता है, वो न्यूक्लियस के पोर से बाहर निकलकर इन अमीनो एसिड्स को कहता है हाउ डू यू हैव टू कंबाइन टू फॉर्म अ पर्टिकुलर प्रोटीन. फाइन, तो डीएनए से इंफॉर्मेशन निकलेगी, डीएनए आरएनए को मैसेज देगा कि तुम क्या करो, तुम ये मैसेज लेकर बाहर चले जाओ और अमीनो एसिड्स को बोलो इस तरीके से कंबाइन होना है. फाइन, नाउ डीएनए मैसेज देगा आरएनए को और आरएनए मैसेज देगा इनको that how do you have to form the proteins, and this is known as the central dogma of biology. Okay, now how is this central dogma made? You have DNA in here, DNA gives information to RNA, and this RNA is actually known as a messenger RNA. It is known as a messenger RNA because it is carrying out the message from DNA to the proteins. Okay, now this is the DNA and this is your RNA. DNA जब मैसेज देता है मैसेंजर आरएनए को, this is known as DNA transcriptase. This is known as DNA. This is known as transcription. This is known as transcription. This is known as transcription. DNA transcription, यानी the message that comes out from DNA to the messenger RNA, the process is known as transcription. The process is known as transcription. और जब RNA, when the messenger RNA carries out this information and gives to amino acids and they form the proteins, that means RNA से प्रोटीन तक ये जो आपका स्टेज है, this is known as translation. This is known as translation. Now, this entire process of transcription and translation, this is known as the central dogma. This is known as the central dogma. The central dogma. Okay, I hope it is clear. What is the central dogma? The message carried out from DNA to mRNA, transcription, from mRNA to protein, translation. That is the central dogma. आपने देखा होगा कभी हम रिवर्स ट्रांसक्रिप्शन की बात करते हैं, RT-PCR सुना होगा आपने. RT-PCR में the message is given from RNA to DNA. This is reverse transcription. This is reverse transcription. So I hope you understand this central dogma of biology. हमने DNA कैसे बनाया, फिर DNA में sequence होती है, that sequence has a meaning, it has a code, and that code is used to create proteins. From that we have cells and tissues and organs in organ system. Now this DNA, the message is carried out through transcription and then the message is carried out through translation. That is the central dogma of biology. I hope you like this session. In the next lecture, I'll tell you how is the chromosome created, okay? From this very DNA that is present in our nucleus, from this DNA, we initially construct, chromatins, and from that chromatins, we create chromatids, and from those chromatids, we create chromosomes, and these chromosomes are, in our body, we have 23 pairs of chromosomes that are a total of 46 chromosomes in our body. So in the next lecture, we'll be covering that. Okay, thank you all for watching this video. We'll be meeting in the next lecture, okay? Bye.